Hello, everyone. Thanks for joining Foresight's Biotech Group today. Um, Today, we're really lucky to have Varun Dwaraka with us today, and he's with True Diagnostics. And today, he'll be talking about epigenetic predictors. So, thanks so much for joining us.
Thank you, Lydia. And thank you everyone for joining on it's 1 p.m. here in the East Coast, but just, taking an hour of your time to just listen to me ramble a little bit about something I've been deeply passionate about for the last 4 years. And so what I'm going to try to hopefully synthesize is this message that goes into this whole idea of using maybe these epigenetic, let's just say, to monitor, personalize, and validate gerotherapeutics and interventions. And before I start, I just want to make sure, I know I just did this whole intro. Is everyone able to hear me okay? Perfect. Okay, awesome. I'm Varun Dwaraka. I serve as the Director of Research and Principal Investigator at True Diagnostic. And I think it's important to maybe kind of, here are my disclosures, but maybe kind of talk about who we are as an organization or an entity if you don't know who we are. So True Diagnostic, what we essentially do is that we're essentially a blood testing company. We have a CLIA certified lab here in Lexington, Kentucky. I am based in Kentucky. And what we do is that we take a lot of the blood fluid epigenome, be able to sequence it, or sorry, quantify it using the Illumina Epic array, and be able to extract information based on the epigenetic nature using DNA methylation as a proxy. And so, as a company, we do have two different branches that we can consolidate to. We have a commercial branch where you can actually take our tests. It is available direct to consumer. But maybe more on the side of where I really exist in is more on the research side. How are we understanding or better understanding these predictors? And how can we maybe use that information to better assess how these predictors, where we can go towards and how to interpret these predictors? And so our research and development team has been around since July 2020. Really, our first step was to really generate a huge methylation data set. We'd like to think it's the largest epigenetic methylation data set in the world. Yeah, but then we've been using a lot of this data to be able to go through and develop new tools and also make that data set available to other like-minded researchers to be able to test their own products as well. And so we have exclusive published algorithms. Either we've published them or we've been able to license them from different institutions. And I think the main thing that I really want to bring this across is a lot of what I'm going to be sharing is based on active research. We are actively researching how to properly utilize and interpret these epigenetic predictors. I know that there are a lot of people out there that are saying things for and against maybe these epigenetic clocks. For us, rather than combating those, let's maybe better understand what they're actually doing and maybe better understand how to utilize and interpret these tools, and then also empower the researchers who are also using these tools and better understanding how to use them. And so through this, we've published plenty of papers, and we've also done this not only by a lot of the work we've done, but really collaborating with the brightest people who have actually shaped our own narratives as well. So let's introduce the problem and why maybe we kind of went into this whole area of epigenetics. So the problem is that when we started as a company, we really wanted to bring information to the average consumer. And so when we started to kind of bring epigenetic testing to the market, what we started to listen to, or what we started to hear, is that, okay, I want to be younger. So this individual who may look like somebody but is definitely not that person, Jaron Bonson, let's just assume that this guy is coming in for his own epigenetic test. And then his, let's say, practitioner, Oliver Dolman, understands, let's try to optimize your regimen. So by this time, he's already understanding what we do as a company. He just wants to be able to optimize and be healthier and maybe extend the health span and lifespan of that individual. And so he starts to utilize all of these different techniques: caloric restriction, sleeping alone, intermittent fasting, all of these things that, has been either mentioned by his practitioner or he's heard from the ether, AKA X. But the thing with all of this is that really what we really want to understand is, of all of these things, rather than blindly following a specific regimen, is this intervention meant for Jared? How do we know that this intervention is actually the perfect intervention for that person? And so in order to do this, just like any other tracker, we have to figure out a way to measure it. And so Gyrin might go through many different clinical trial or the clinical outputs, or just look at different tracking mechanisms. So you can look at musculoskeletal changes, serum markers, inflammatory markers, stem cell changes, et cetera, et cetera. Now, oh, yes, I am sharing slides. Oh, I apologize. Oh my God. Okay. Does this make sense? No? Okay. Okay, yeah. Here's Jaren for those of you guys who are listening. Anyway, so with Jaren, he might go through all of these different panels, but the thing that we wanted to really show here is that the current state of testing really focuses on a few things that are maybe not as forward-looking. They're more, for example, a lot of the present health standards are really based on standard physical exams, and they really lack insight into the long-term biological processes. Some of these tests are reactive by nature, so their traditional testings are designed to really detect existing conditions, but only after symptoms and damage have occurred. They also work in snapshot approaches. So essentially, they're going to be able to capture maybe more momentary pictures of health, but they offer little information into how that lifestyle and environmental factor really shapes health over time, and also limited aging insights. So these tests don't really assess cellular aging or how well the body's adapting to the stressor. And so with all of these different aspects, we started to look into maybe different ways of capturing it. And this is where I think initially, as a company, we started entering into this whole area of epigenetic testing. Just as a basic biomarker, epigenetics really focuses on maybe a little bit more on the side of gene expression. So where if you have areas such as the CPG island, which are methylated, so physically methyl groups are being added to the cytosine of these islands, you can actually facilitate typically the gene that is associated with that CPG island to be turned off or on, depending on that methylation or unmethylated perspective or orientation. Now, with this said, with this basic understanding, what we start to realize is that you can actually go beyond the surface. So with epigenetic tests, while physical exams and blood tests really measure the current state of health, epigenetic testing can identify underlying biological processes that influence long-term health and aging. And what's more interesting is that with the epigenome is directly influenced by those environmental cues. So a lot of what you're capturing are signals based on changes that are environmentally that are occurring at that to that individual environmentally. And so this is what was really interesting. And we started to realize that there was a gap in the tests that are available that really capture the epigenetic changes that are maybe capturing more of those environmental cues compared to something like a genetic test where a lot of your genes and your DNA are very much static throughout your entire life. We also wanted to focus a little bit more on the proactive versus reactive health insights. So epigenetic testing really does provide early risk prediction, enabling preventative action before those diseases develop. And what I'm showing you here is actually some results from a clock that I'll be introducing later in my talk, where essentially these AUC curves are essentially looking at the 5-year survival and 10-year survival prediction. And what I hope you can appreciate is compared to just your chronological age, many of these epigenetic predictors, such as these epigenetic clocks that I've identified here, are showing higher predictive ability compared to something just as age, which age is still used in the clinic as a potential prediction of disease manifestation. And something like this can be converted into a potential disease risk percentage that then can be given to the consumer or the patient in question for not only just all-cause mortality, but can be stratified based on different disease types. They're also very dynamic and adaptive. So because of that ability to capture more of those environmental changes, those epigenetic changes actually reflect a lot more of those environmental factors, and thus they can actually change based on exposures, based on different types of environmental cues. But then also using this is actually an example of an individual who has been taking our tests. We could see how, for example, different types of treatments can really influence those outputs as well. So these are 2 different clocks that we offer. And this is basically really showing and honing in on the dynamic and adaptive nature of these tools. And then finally, it really uncovers more hidden information. So epigenetic testing can reveal critical insights in how your body is aging and adapting that which traditional tests typically miss. You might be wondering why we really focus more on aging than let's say individual diseases. Well, aging is one of the leading causes for the progression of many of these different diseases. So if you can capture that aging process better, maybe that could give you a lot of insight into the manifestation of those diseases as time goes on. And so with all of these different questions, these different observations, the solution that we started with was to offer a test which assesses your epigenetic states and calculates those epigenetic clocks. So not only just looking at your epigenetics, but we started with those clocks as a first output. Now, you might be wondering why we focused on epigenetic clocks. Really, this whole concept of epigenetic aging was, I think, really popularized by Horvath's initial clock in 2013. And as time went on, these clocks just got better and better. And what we started to notice is depending on those clocks, you can actually look at the value of the clock in relation to the person's chronological age, so that age based on the date of birth, and see if that value is lower than the chronological age or higher than the chronological age, which really reflects the age deceleration and age acceleration based on those biological outputs. And what was really interesting is that actually this is a very good review where those decelerations really correlated with better outcome based on, for example, healthy food intake, exercise, physical fitness tended to decelerate that age, whereas those age accelerations not only correlated with really good outcome, so for example, really bad outcome. For example, cancer risk was really associated with higher age acceleration, stress, neurodegenerative diseases, but you could use this as a proxy to look at how well your cellular age is actually present. And so with this said, the next question came to what clock do we use? So this image right here is purposefully convoluted. And the reason why is because when you start looking into this area, into this field of epigenetic aging and all of that, you start to realize there are plenty of clocks. But the best way to maybe think about this are maybe in generations. So for example, a lot of the first generation clocks, which include the Horvath clock, were really trained to chronological age, where you were essentially identifying which CPG site or DNA methylation locations were actually predictive of that chronological age. As time went on, people started to realize that maybe training to chronological age or the thing that that you're trying to get away from is not the best case. And so what people started to do after, so for example, with PhenoAge, is start creating a phenotypic score or using current estimators of overall, let's say, morbidity and mortality, and then training those CPG sites to those outcomes as well. And these are generally referred to as generation 2. Now, when we start to get into generation 3, this is where there's a little bit of a deviation. But for us, we call generation 3 as those clocks that are actually trained using aging phenotypes longitudinally. So an example of this would be Dunedin Pace, where they actually followed a cohort of individuals from Dunedin, New Zealand, where the name comes from 1972 all the way to 2018, and essentially created a pace of aging score. So compared to something like a biological age clock, this output is essentially going to be a rate of aging or a pace of aging, where 1 is the rate of age accumulation per every year. So any pace of aging that is less than 1 would essentially mean that they are aging at a slower rate compared to something over 1, which means they would be aging at a faster rate than every year that has elapsed. And so when you start to think about this, you have to also realize that each individual clock is not going to give you the same answer. And the reason why is because the differences in the clocks are essentially based on how they were trained. So this preprint by Morgan Levine, who actually created the original PhenoAge, did a very good job kind of going through what makes a clock tick, no pun intended. If you look at some of this figure right here, what each of these different color modules represents are groups of CPGs that are similar between each individual clock that is represented in this example. And so what I hope you can appreciate or notice is that some clocks have very similar color schemes, for example, Grimage and Dunedin Pace, but the amount of those CPGs or those features, let's say, will deviate based on individual clocks themselves. And so because of this, the takeaway that I hope you can take away from this is that each clock is capturing something different and explains why you get varying outputs per clock. We've heard a lot of people say, this clock is showing this, clock is showing this, thus none of them are agreeing. But really, that is kind of true sometimes. It all depends on how the clock was built, and thus it is very important to consider what that clock is actually capturing based on how they're developed. And so for us, we had to make a couple of choices. We had to go through different criteria in order to be able to choose individual clocks. So the clocks that I'm going to show here are just examples of what we think are the gold standards. And so the five things, or let's say 5 things that we really considered were: are they published? Are they correlating with quality of life outcomes? Are they precise? Are they responsive to interventions? And do they explain why you're aging? These first 3 actually were described in my first Foresight presentation. So I'm not going to go through those 3 per se. Feel free to go back to that presentation, but I'll focus maybe more on these 2 because since that presentation, these were the 2 things that we've actually done a lot of work in really understanding. So the first one is explaining why you age. So since 2023, when I first gave the talk, there were 2 clocks that came out during that time. The first one is Symphony Age, which was done by our collaborators at Yale University, August Segal and Albert Higgins Chen. What they ended up doing was actually being able to construct a epigenetic age that is based on different organ systems. And while it can be synthesized into one single age measure, so imagine you had an age of 43, they actually found a way to deconvolute it so that 43, which is the overall, let's say, age of that individual, you can now look back and say, of the blood samples that were collected, how many of those biomarkers associated with the lung system are actually showing an age of 38? What is the age of the metabolic system, musculoskeletal system, and blood age system as well? This way, this deconvolution at least gives a little bit of an explanation of why that overall composite age is 43, and it also gives a little bit more information on what areas to pinpoint for maybe some interventions. Similarly, we've also at True Diagnostic, in collaboration with our partners at the Mass General Brigham, we're able to develop omic MAge. A lot of these clocks are really taking into consideration DNA methylation, but we also recognize that DNA methylation is a single part of the epigenome. And beyond that, we also have transcriptomics, proteomics, metabolomics, and genomics to consider. And so in order to be able to synthesize all of those signals into one measure, we actually created omic MAge. Actually, my first talk with the Foresight Institute was describing this, so I won't go into too much detail about how we actually created this. But the best thing that I can maybe explain to you is that by synthesizing all of this information, we were able to get one single age measure that seemingly was able to take into consideration all of these different layers of the multiome. But when we go back to this whole understanding of explaining why your omic MAge is high or low, one of the things that we were able to do was actually use the different composites, because the way that we trained this clock was actually creating models of different clinical outputs, metabolites, proteins from the DNA itself. And so by doing so, you can actually use the DNA methylation data to essentially estimate what their clinical values and their protein measurements and their metabolite measurements were. And then using that, you at least had a little bit of an explanation as to why that omic age would be higher or lower. And so with that, we seem to have some, tools or some strategies for explaining at least a little bit of why you're aging. But the next thing, which was the hardest part, is understanding whether these clocks were even responsive to interventions, and also which interventions were changing these clocks, and also which direction as well. And that took a little bit more time. So to start this, I'm going to maybe start with one of the more preliminary meta-analyses that we did with our partners at Yale. This was a preprint that came out, I think, about 6 months ago or so, where we partnered with Raghav and Albert again, and pretty much provided data from different interventions that we have done with different physicians who take our test. And what they essentially wanted to do was really understand which DNA methylation aging biomarkers were really responsive in these 51 different interventions. So I will say, if you want more detail, definitely go to this preprint because there's a lot of information. But I think the summary really is part of this figure right here, where essentially we were able to look at 51 different interventions, and we were able to see as a meta-analysis how if you used all of the different clocks, which ones were really lowering the overall biological age and which ones were increasing the biological age with that intervention. So many of these interventions were not cross-sectional, they were actually longitudinal, but we were able to see, just from a general perspective, what was really decreasing or increasing those epigenetic clocks. So just as maybe a like overall summary, there are a lot of caveats I'll go into, but the top five that we start to see that were lowering a biological age was antiretroviral therapy, anti-TNF alpha therapy, metformin, HBOT, high pressure, and AC11 supplementation. The first caveat I do want to say is that many of these different data sets were done on individuals that were actually undergoing therapy because they were already morbid or that they had comorbidities. So, for example, a lot of these ART therapy and the anti-TNF alpha therapy, these individuals were already very morbid, and so thus if they're already showing aspects of disease, there's a reason that could be a confounding variable for why their overall epigenetic age is decreasing at a much faster rate than something like, for example, HBOT, which is hyperbaric oxygen treatment, where many of these individuals were just normal or healthy individuals.
Yeah, like for instance, you have kidney transplant and gastric bypass in there, which is not something, those are for morbid people.
Yes. Thank you. Yeah. So, I mean, we wanted to be able to do more of just a general way of being able to look at all this information. But that is one thing that, as you're reading this paper, just to keep in mind. But what we really wanted to understand was which of these clocks were actually responsive. Even in this case, this effect size that's being shown is actually a meta score of more than 110 different epigenetic biomarkers, so different clocks and different EBPs and so on and so forth. And so when you start to so it's great as a summary tool. And using this, we were able to even look at different types of lifestyle. So we were able to segment in different types of interventions based on if it was a lifestyle intervention, so diet, exercise, if it was a pharmacological intervention, such as metformin, rapamycin, ketamine, so on and so forth, supplement or medical procedures. If we did the meta score, what we noticed was that lifestyle and pharmacological scores, the interventions, had more of an impact on these overall meta scores of these epigenetic clocks. But I think the most important thing here, and kind of goes back to, I believe, Morgan Levine's preprint, is that it really does matter what type of clock that you're using. Because, for example, if let's just say you use something like systems age, you'll notice that it's not, for example, certain of the components of system age, which includes metabolic age and musculoskeletal, seem to show improvements across all of these different lifestyle interventions. So the interventions are listed on your x-axis. But that's not the case with other clocks as well. And what we see is that with many of the other clocks, some of the clocks are showing improvements across all different types of interventions, whereas some of the other clocks are actually a little bit more pinpointed for particular interventions. And so it's important to remember that the type of clock really matters. And in that paper, we also segment based on the different clocks and really shows that the generation 2 clocks, which I'll explain, are doing a better job at capturing these interventional changes. But one of the reasons why I wanted to put this slide up is because the most important thing out of this is that rather than just using those composite clocks, for example, Dunedin pace or systems age by itself, these explainable clocks really give a little bit more resolution. So while systems age may overall show a significant decrease, by deconvoluting into these individual clock types, you get to see really more of a story based on, what is actually being improved if, let's say, you do a low carb diet or a Mediterranean diet. Based on this result, we would expect to see a greater decrease in the metabolic and musculoskeletal clocks specifically. So one could make the interpretation that these diets were really affecting these particular systems. And so going back to this whole concept of the clock really matters, or predictor even really matters. This figure right here is actually kind of getting to that, where what I hope you can see here is that you're seeing the same type of effect size across different types of clocks. And some of the clocks are showing significant decreases while others are showing significant increases. And so this is where it really goes down to, what is the clock actually showing? How was it developed? And is it capturing the signal of interest that you're really interested in? In the case of the lifestyle and the pharmacological, one thing was pretty apparent to us is the clocks on the right side, which typically represent more of those generation 2 plus or generation explainable clocks, so generation 2 or higher. Those clocks tended to have more concordance with the pharmacological and lifestyle changes across the board. Whereas some of those generation 1 clocks, like Horvath, actually show the opposite, which was something important to really identify, and really goes to show that whenever you're using these tests, maybe for more intervention tracking, generation 2 or higher might be the better way to move forward. But more than anything, it really reinforces the importance of looking at multiple clocks. For us at True Diagnostic, we've decided to, maybe more to our detriment, to provide more data than typically out there. And the reason why is because we think that each of the different clocks, like for example, Dunedin pace, systems age, individual components of systems age, and omic age, are all telling a different story. And it's better to look at each of these data points separately rather than consolidate it into one metascore. But the reason why I wanted to kind of start with this is because this kind of also takes into consideration some of the papers that we've been able to publish. A lot of the data that we provided for the 51 intervention data were data sets that we've actually collaborated with different physicians or even research groups. So for example, when we start to look at, let's just say, this vegan diet group, this was actually part of a study which we published back in 2024, where we were looking at a set of twins that started off as omnivorous, but one set of twins went into a vegan diet versus the other set of twins ended up going into an omnivore diet. And so this is actually available through BMC Medicine, so feel free to take a look. But we did this on 21 pairs of omnivore twins, and we applied a lot of these different epigenetic predictors because we were able to get blood samples from these individuals at baseline, and then at 8 weeks after the treatment. It is also important to say that this was done as a randomized control trial, and the comparisons were really done between the vegan versus the omnivore, both comparing within each cohort, comparing 8 to 0, and then also between the 8-week versus 8-week. And what we ended up seeing was quite interesting. We saw that many of these different clocks were actually showing significant decreases across multiple types of clocks. And many of them were actually ones that we didn't develop. These were developed by other academic institute or groups and different institutions. So the pace of aging, pheno-age, grim-age, and many of the systems age seem to show significant decreases, whereas telomere length, this was done based on quantitative PCR, actually showed an increase once these individuals were on a vegan diet. Versus those individuals that were on the omnivorous diet showed no significant change when comparing 0 to 8 weeks. Now, this is not to say that you have to switch your diet or anything. More than anything, this was a methodology in which we just wanted to see what signals of a healthy vegan diet or an omnivore diet is able to be captured by these epigenetic clocks, which we started to see was many of these generation 2 and generation 3 clocks.
Question?
Yeah, Krihan.
That's a rather, how can I say this, low resolution view of the data, a minus or an NS. Like you say, okay, so the direction of change, I mean, how much within the noise sampling level, or what are we talking about here?
Yeah, so I had to maybe minimize it just for the sake of this presentation. But in the actual paper, we have the bus to show. The reason why we considered them to be changing is that they actually achieved a significance threshold of at least a 0.05. And the direction was a significant change. So I didn't want to say anything more than, how many years of increase or decrease, because we also converted these into age accelerations before actually doing the statistics. But everything that has been shown here was a significant change based on a p-value of 0.05 in the direction of negative or non-significant.
Thanks.
And I believe this figure was figure 2 in the manuscript. And more importantly, so while we can also look at these ages, these EBPs actually served another, were able to offer more greater resolution. And these EBPs, these epigenetic biomarker proxies, are essentially the components that make up omic age. Now, I think you'll notice that omic age was not significant in this slew of significant clocks that changed. However, when we actually looked at the EBPs that were changing, that were part of omic age, we started to see that many of these different EBPs were showing significant increases or decreases. Now, again, to Creon, good point. I didn't show what the p-value change was here, but that was only for the purpose of being able to put this onto a slide. But many of these actually passed an FDR threshold of 0.05. And so the direction, which is the direction of at 8 weeks versus the 0 week for the vegans and the omnivores, were identified here as well.
Since we don't know, other than maybe for some obvious things in telomeres, which direction is the good direction? What is that? The bold face is bad direction, or how does this work here?
Yeah, so that's the thing. So right now, these EBPs, we just identify and we're able to show in the paper. But one of the things is we don't necessarily know, like many of these, for example, this sterile glycerol, we don't know what its impact is in a vegan diet, or at least at the time of writing, we didn't know. And so this is more of identifying essentially proxies of these different metabolites, proteins, and, for example, clinical markers that actually were, maybe associated with the vegan diet. For example, triglycerides, I think we could say that a lower triglyceride at 8 weeks is probably a good thing. And that's what we observed here with the vegan diet, which was not reciprocated in the omnivore EBPs. And so this EBP, or the epigenetic biomarker proxy for triglycerides, seems to show an improvement in the triglycerides at 8 weeks. But for the other ones, this is, again, it's more of a hypothesis building tool or a panel to really assess a lot of these interventions based on these DNA methylation data.
Question.
Yeah.
What do you know about this? I've never seen Margaroyal GPE.
Yes. So the one that I did include is the next slide, which is ergothioneine. So for example, ergothioneine is associated with reduced mortality and decreased risk of cardiovascular disease. In the vegans, it's actually shown to be increased versus the omnivores being decreased. And so in this paper that we actually, again, using just this data, we went back to the literature. It's essentially a metabolite which is strongly connected to health conscious food patterns and was associated with lower risk of coronary disease.
So ergothioneine is this Margaroyal GPE thing?
So this is going to be one of them. But for one Margaroyal GPE, there's actually not much literature associated with one Margaroyal GPE, especially in the context of vegan and omnivore or just diets in general. So it does actually, it's a great point to bring up that this also allows you to do a little bit more of a, let's say, hypothesis building of now what does one Margaroyal GPE do in the context of diet and moving forward.
Yeah, exactly.
And actually, yeah, so that's kind of the slide that shows here is right now, like when we looked at it, it really has no association to diet. We just know that there's 2 SNP variants that are associated with it. And so that connection between one Margaroyal GPE and then also the SNP that's associated with, let's say, reduced risk of advanced AMD, we don't know what that link is, but it's now something of interest, which now you can use. By using those EBPs, now we were able to kind of get to a point of maybe highlighting this as a metabolite in question.
It sounds like it could be a membrane fatty acid, or 17 carbons saturation, 0 saturation. So, and in AMD, you have a lot of free radical oxidation. It could be part of the retina, membranes. Anyway, that's cool result.
Yeah. It's especially from a data-driven approach, it's kind of an interesting signal that we observed, but we'll let people that are a little bit more, let's say, smart in this area or intelligent in this area to kind of maybe handle the interpretation. And we did this also with another study that we actually put out as a preprint, and it was recently accepted into a translational psychiatry. And so in this study, we actually it was using ketamine treatment on individuals with that were already identified as having PTSD or MDD, and seeing how the ketamine was able to, let's say, decrease or increase their DNA methylation, the epigenetic clocks, or the different biomarkers of aging. And okay, it's supposed to have a slide of all of the different clocks, but essentially the clocks that we looked at were omic age, grim age, pheno age, Dunning pace, and a slew of other generation 1 clocks. The reason why I highlighted those were that in these individuals, the omic age, grim age, and pheno age showed significant decreases after 2 to 3 weeks of ketamine treatment. And again, these individuals were already pre-selected for showing PTSD or MDD clinical scores, or characterized as having those 2 different comorbidities, using the PHC-5 or PHQ-9 clinical scores. And one thing that this manuscript really showed, which was really interesting to us, is that we weren't able to capture any of those changes in the clinical scores in many of the clinical lab panels. So for example, they had full clinical panels done, which included CRP, IL-6, and a slew of other, let's say, lipid and protein panels. And none of them showed significant changes. Really, the only ones that showed changes were their PCL9 and PHQ9 and PCL5 scores, and the omic age, grim age, and pheno age clocks, which really got us thinking maybe there's a component of mental health that these epigenetic clocks are really capturing, that maybe some of these clinical, let's say, inflammation panels aren't able to recapitulate. I do apologize that this slide was removed, but I'm more than happy to share this preprint during the Q&A. The other thing that we also looked at were specific, let's say, supplements. And so this particular study that we put out, the only thing that I really want to highlight is, again, something that you guys might already know, is that whenever you're using these different types of tracking mechanisms, it's really important to not just look at the population level states, but also go into each individual person. So each of these panels are showing the PC pheno age value across individuals that were part of this intervention. And what I hope you can appreciate is that not every individual is responding the same way to their clocks to these different supplements. And so it really, one of the use cases that we've always been kind of pushing for the use of these epigenetic clocks is very similar to maybe any other tracker. You have to be able to look at each individual to show the heterogeneity of the response. And it's a great example of how you could use these tools in an N of 1 case study, especially for clinicians moving forward. I think the one good example is kind of what I showed you before, where you can actually start to track many of these changes and say which intervention was being done at a specific time, and then see what the response pre, during, and after of these different clocks, how they respond based on those interventions, which can give you a little bit of a better understanding of how that intervention is really affecting that individual.
Question?
Yes, Creon.
Don't we have, I realize that you have these clocks, including your multi-omics clock, that try and sort of summarize or all these separate little into, how is this affecting my biological aging? But isn't it the case that we look at one of these things that's going way down in this chart here for a given person, that, we don't want to over-index on this and say that the guy that goes down means we're really being successful, because for all we know, in isolation, that thing going down may not be great for aging.
So is the comment based on, is the decrease that we're seeing actually important, or is it actually a positive for aging in general?
When taken in isolation, like the problem is, don't we need to know that, like it might be that, just arbitrary, it might be that if, these 2 indicators go down and everything else stays the same, that's not good. You have to have these 2 indicators to go down and these other 3 go up and these other 3 unchanged. That's good.
Right. Yeah. So, I mean, I largely agree with you. I think that really understanding what these clocks are showing is actually the biggest challenge. A lot of the times, the clocks that we're selecting for, and if I had more time, I would probably go deeper into these clocks. Maybe the associations to disease are actually one of the indicators of whether, for example, pheno age or grim age or Dunning pace are actually clinically reliable measures of aging. So, for example, we know that with decreases in pheno age, you actually start to show, decrease in your pheno age, your overall, let's say, disease risk also starts to decrease as well. And so based on those kind of measurements, we are suggesting that the decreases in pheno-age are actually good because of that association to particular diseases.
Okay, let me put it differently.
Yeah.
Maybe you've already got this, but wouldn't it be useful to have a chart? And this is something that can be essentially obtained just from the definition of the pheno-age, or omics age, not from individual data. Wouldn't it be good to have a chart that first of all shows all of the markers in isolation and how much of a beneficial effect it has if that marker moves a certain amount in a certain direction, and then another chart that shows like pairs of markers and shows what if the pair moves like this and that, how does that affect the overall omics age? Because you kind of want to watch out for just someone might have some interaction that really pushes hard on one of these markers and might even therefore improve the clock, but it might not actually be good for a person.
Yeah, I actually would agree with you. I think so. The idea of using not just focusing on just, let's say, pheno-age, but then having other markers as well, omic age, deneen pace, whether they're also showing similar changes as well.
Okay.
Yeah, I will say when this paper came out, we applied a few clocks, but not all of them. I think that, for example, what we did here might be a better method in maybe showing whether it's actually expansive beyond just one measure of the clock. So I think to that extent, I would agree with you that maybe looking at other clocks here would have been better. But I do think that for the point that I was making here, I think it's just more of, using these in the N of one. This is kind of an example of, what you could do with just, let's say, pheno-age. But I do agree, having a similar chart with omic age, with grim age, deneen pace could also show you if, that individual is responding systematically to many of these different aging markers.
Thanks.
Right. Okay. And so I think with my current slides, they were a little bit jumbled. So this was actually the data set that I wanted to show for ketamine. So the data that we showed are PCL5 and PHQ-9. So these are different scores for PTSD and MDD. And then omic age and grim age are also showing significant decreases at post-treatment, which are consistent with what we see with these clinical scores. And just to reaffirm this idea that these epi clocks picked up a signature that other clinical variables did not. And more importantly, because we saw a change in omic age, we were really curious as whether these EBPs, which make up the overall omic age quantification, which ones of those changed. So again, kind of similar to what we were talking about before with one Margolyl GPE, many of these are still pretty are not really investigated as much. For example, we know that elevated adenosine levels associated with enhanced neuroprotection, reduced inflammation, modulate excitatory neurotransmissions. We know this based on the literature, and it aligns with ketamine's therapeutic actions in depression and PTSD, but this connection hasn't really been made before, especially in the context of ketamine. So this really brings up another methodology in really building these hypotheses. And then the other thing that I'm going to talk about very briefly, because I realize I'm running out of time, is this whole usage of these epigenetic clocks in just generally in overall pharmacologics. So ketamine was an example of it, but there was a recent study that we were affiliated with, which was using these epigenetic clocks as a tool to assess impact of semaglutide in HIV afflicted cohort. So one of the things, there was some press that came out with this particular preprint. And one of the things I do want to mention is that these individuals that were part of the study, because we were able to get the data, they were part of a phase 2b double-blind placebo-controlled RCT, which were using HIV patients that were on stable antiretroviral therapy. So there's a lot of, let's just say, asterisks with the population, but we needed to get started at least with an RCT just to see how these clocks would maybe change. And so the manuscript is actually available on bioRxiv. And so one of the things I just want to maybe go into are the results. So there's a lot of information right here. I think when this video comes out, feel free to pause and maybe look a little bit deeper. But I think the thing that we've noticed immediately was how similar to, I guess, what Kriyan, you were mentioning, is how many of these different clocks were showing decreases in their overall group difference in the semaglutide cohort relative to the placebo cohort. This was also recapitulated with Dunedin pace as well. So not only are these different epigenetic clocks that were outputting overall ages, but even the pace of aging was significantly decreased. Now, one of the things that we're actively working on is now really the big thing that this is kind of telling us is that there's a signal with a lot of these pharmacologics that DNA methylation is capturing. What we wanted to do is to just see if we can utilize any of this information to maybe develop companion diagnostics. So essentially, could we develop a test that would tell you if you are effectively going to change your epigenetic age by using, let's say, a ketamine or semaglutide, or let's say a statin or any type of drug?
Okay, so statin's going to be interesting because they have all sorts of side effects. Now, this semaglutide thing, in your much earlier chart where you showed all the different interventions, it was like a box plots or something that were sort of
the effect sizes. Yeah.
Yes. There, semaglutide didn't have much of an effect. Does that mean because it was not like, is this semaglutide if you have HIV, or is this, how relevant is this to those of us who don't have HIV?
Yeah, so that data set was actually a short-term data set on healthy individuals versus this is a data set that was on specifically HIV patients that were undergoing ART treatment specifically. So they were actually two different groups. This was a completely separate group as to that one. Now, the reason why in that data set we're still actively going through that data set is because we wanted to first look at the RCT, which this is the data set for that. And we're going back to see what are the differences between why this, cohort was showing changes versus, let's say, a healthier cohort. So that data hasn't been made public yet, mainly because we're still investigating, just making sure we didn't, leave anything under a rock per se. So with this said, I'm not going to talk about the statins during this call or this talk. But what we did do is just as a proof of concept, we wanted to see if we can create a paired companion diagnostic. And so what is being shown here are essentially ROC curves of two different companion diagnostics that were trained to essentially BMI. The way that we're doing this is that we're defining a binary responder label from the baseline. So are we seeing a change of 5%, either increase or decrease, a 5% decrease in your BMI, and considering them to be a responder within the semaglutide arm. And so using that information, what we could do is train from baseline a predictor of whether we could predict if an individual is classified as a responder or non-responder, and then validated it on a placebo arm, which was part of that second arm in the RCT. And so what we're seeing here is this is unpublished data, and we're still working on this. But the reason why I'm showing this to you is that there's some type of signal that we're capturing, which is able to differentiate between those responders of GLP-1 versus the placebo group. And now what we're essentially doing is now better understanding what those CPGs are that we've been able to capture moving forward, and what that means to the overall ability of classifying semaglutide intervention or not. And so there's going to be a lot more work needing to be done. This was actually, I think, as of 2 weeks ago, this was data that I was able, or results that I was able to come up with. But as we move forward, any kind of advice or any suggestions would be very helpful as well.
Here's a suggestion.
Yep.
You haven't already done it. Collaborate with Greg Fahey and see what his intervention, whatever it's called, TrimX, how it stacks up.
That actually is a fantastic suggestion. We just haven't sent that email yet. So I think that's something that we see eye to eye on. All right. Now we have 4 minutes, and I was going to go through this whole idea of how we're redeveloping the epigenetic biomarker proxies, but I know that this has been a lot of information so far. And so with all of this said, I'm more than happy to share some of these, slides with you all. But in, I guess, the last 4 minutes, I just wanted to maybe stop there, and we can maybe discuss a little bit more about, what are the future next steps? How are we maybe improving upon some of these epigenetic biomarker proxies? And then also maybe comments, suggestions. And if anything, the last thing I really wanted to point out is this right here. For a lot of the work that we've done and a lot of the work that I've shown here, this has been an entire consortium of individuals who, people that we've read their papers and we've collaborated with. And so we have been working to make a lot of these tools a little bit more, not open source, but available to researchers that want to maybe publish and better understand how they're working. And so if you're interested in utilizing our models, and even if you want to run biological samples at our CLIA certified lab, feel free to reach out. We are more than happy to accommodate any of these requests, and we'd love to, co-publish. We'd love to, really bring some of these predictors out into the world. So yeah, please contact us. And that'll be the end of my talk. I'll just leave this up.
Thank you so much. I guess what we could do is open the floor for one burning question from anybody if you haven't had the chance.
If I could ask a question. I think the basic question is to what extent you can actually make epigenetic results actionable. Obviously, we know that playing a 90-minute football match really has a good effect on your epigenetic age, but I don't go for football that much. And I recognize that all biomarkers have problems, but how can you actually make an epigenetic test really actionable?
Yeah, no, it's a great question. And I think that, sorry, let me just put okay. Yeah, so I guess during grad school, a lot of the actionable aspects of these different biomarkers is really based on, let's say, knockout models, especially coming from, genetics and transcriptomics. You take away that particular thing and see what the response is. In certain cases, there was a paper that recently came out where they used CRISPR-Cas9 on individual cells and actually modulated these particular CPGs that were part of these different clocks. I believe the one that they looked at was the skin and blood clock for Horvath. And what they started to see was that based on the individual CPGs, they were able to either modulate or decrease the clock, and also showed changes in the overall cell type, just I believe it was the look and size and the health of that particular cell. And so in order to look at those actionable aspects, that's one way that you could really start to affect it. But I think for us, actionable is really maybe looking at these interventions to show how these interventions are maybe increasing or decreasing these epigenetic clocks, and how consistent are they with, let's say, the known lab markers or the biomarkers that you're already using. But I think there's still more to be learned about this. And the way that we have attempted to address this is through the interventions. But as time goes on, with especially those modulations of the CPGs, that's where you can start to maybe better understand how these clocks are actually working. And then also, how actionable are these clocks in the context of other estimators of health.
Yeah, but at the moment, they're not actionable.
In the moment, I think that we are seeing, for example, I think the vegan and omnivore trial that I presented on, it tends to show that there is a response that these clocks are changing. And also with the ketamine trial, or sorry, not the ketamine trial, the vegan omnivore trial, there's another paper that came out with all of the clinical outputs, like, for example, the inflammation panels and those markers. And we're seeing consistency between the two. So the actionable aspect could be that there are specific interventions that seem to be decreasing those clocks. And one could argue that is potentially a form of actionable outcome that you could work on.
Well, we disagree.
Okay. All right, then. I think we're at time. So thank you so much, Varun, for joining us and for the great presentation and for fielding all of these questions. Thank you all in the audience for joining. One thing before we leave, I just want to give an announcement, a little housekeeping. We are all going to be meeting at the end of December for Vision, or at the beginning of December for Vision Weekend. It's going to be December 6th and 7th. And we'll have a lot of really amazing longevity speakers in attendance. So we'd love to see you there. It will be in San Francisco. And with that, thank you guys so much, and hope you have a great rest of your day.